All right. Good afternoon, everybody. Welcome. Thank you for joining us here on behalf of all of us at Virtual Force Space. Welcome to today's event, Microorganisms for Sustainable Food from Earth to Space. We are very happy to welcome back Concordia Université Laval iGEM teams for organize this event, as well as today's moderator, PhD candidate in synthetic biology, Kenza Samlali, and of course, the special guest presenters who will be introduced shortly. Before I pass the mic to Kenza, a few tips for our audience. If you have questions throughout the session, we invite you to post them in the Q&A that you see at the bottom of the screen, and Kenza will attend to them in due course. And of course, if you're watching on Facebook, feel free to comment there, and we'll make sure to bring your comments and questions to our panelists here. So it's now my pleasure to briefly pass the mic over to Kenza for a quick introduction. Hi, Kenza.
Hey, Anna, thank you so much. Yeah, so we'll actually start with the two iGEM teams who will introduce them. They are organizing today's event. So thank you so much also for inviting me to moderate today. So I'll give the word to Angel from ULaval.
Oh. Right. Yes, I'll start and then maybe I can follow up. So yeah, hello everybody, we're iGEM ULaval, and we're working on a project on helping repurpose defective maple syrup for different purposes in the future.
Yeah, so thanks to talking to experts in the field and producers, we decided to work on ropey maple syrup, which is a product that's thrown away in this industry. To add value to ropey maple syrup, we're creating a biological treatment that transforms this product into a base that resembles maple syrup. If you want more information about our project, stick to the end of the conference where some of our team members will be here to answer your questions. Now we're just going to pull up a poll that talks about your level of expertise in synthetic biology.
Great, thank you so much. So as you see, we're popping up polls sometimes, and that is mostly because the iGEM teams are also using this kind of for having feedback, and they're organizing this and as part of their competition as well. So next we have the Concordia iGEM team. You want to introduce yourself, Paula?
Hi, hello everyone. Yes, my name is Paula Gomez. I'm a team member from iGEM Concordia. This year we're working on Asteroids, a project that aims for space food production by making yeast compatible to space. We organized this event thanks to the support of Sustainability Action Fund and For Space Concordia. This event is part of our science communication aspect of our iGEM project. We will be posting polls to see the impact of this event, such as the one you're receiving right now.
Thank you. All right, I think there's another poll popping up just to evaluate a little bit our audience, and it will also help me guiding the questions from our speakers. So if you'd answer that, that would be fantastic. So now we will continue with 3 talks from really awesome speakers. If I can ask all of you to post questions in the Q&A. So on the bottom of your screen, you will see a Q&A button. Here you can leave your questions and we will select good questions and we will try and answer all of your questions. So I hope there's a lot of engagement here with the audience today. Another thing I want to ask you is, someone can pull up the slide again. You should change your name if you want to. So if you go to the 3 dots above your name, you can change your name. And if you would please change it to a format where it's your name, your pronouns, and your affiliation, then it will be easier for us, yeah, you can see it in the video here, to address you correctly. So another note, yes, as I said, leave questions in the Q&A. After the first 3 presentations, which will be just really short presentations, I will address maybe one question in case something is not clear. And other questions will be used during a half-an-hour Q&A. So let's get started. And we will start with Morgan Irons. She is the founder and chief officer of Deep Space Ecology. And this is a space and agricultural business startup working to solving the challenges of food security on Earth and deep space as we venture out to our solar system. So this is actually quite a remarkable startup, I would say. So her work concentrates on sustainability in extreme environments and long duration space habitation. Morgan is at the same time also a PhD student in soil and crop sciences at Cornell University. And besides his science work, she does a lot of outreach as well. So please, Morgan, if you want to take over, you can share your screen, and I'm excited to hear your talk.
Wonderful, thank you so much. So let me share my screen. Okay, there we are. Okay, hello everyone, my name is Morgan, and just like Kenza said, I'm with Deep Space Ecology as well as a PhD student at Cornell University. And so a lot of my work focuses on not only Earth agriculture, but also space agriculture, the entire system from the soil to the plants and how the plants interact with the atmospherics and the surrounding technology as well. And why is it so important that we start talking about space agriculture and have these conversations? Well, especially when it comes to Mars, we see things happening. We see that NASA had their Authorization Act of 2010 where they want to send a human crew by the 2030s. And SpaceX, of course, wants to be sending cargo by 2022 and then a human crew by 2024. So having these conversations and doing this research about how we'll actually be able to feed the crew and how we'll be able to actually develop agricultural systems for more long duration space missions and eventual long-term habitation on places like the Moon and Mars is something that's very important. And it's important to also realize that this conversation isn't necessarily a new conversation. There's actually been a lot of research being done on closed ecological systems for space habitation. Some of the earliest mentions of closed ecological systems for space habitation was actually in 1895 with Konstantin Tsiolkovsky, who published the book Dreams of Earth and Sky, which first details the use of plants in space exploration. And Tsiolkovsky, you may recognize the name, is also considered the father of rocketry and space exploration. So he was also a literary figure, interesting. And so you see that this research has been going on from a while. A lot of it started in the 1960s in the Soviet Union with the BIOS experiments, and then NASA would start having conversations later on in that decade talking about bioregenerative systems. And moving on, just really continuing these conversations, continuing these experiments. And we start to see other countries getting involved as well. Japan also gets involved with constructing their closed ecology experiment facilities. Canada also has universities and researchers involved in closed ecological system research. The European Space Agency with the development of their Melissa system, and then really recently China with the development of their Lunar Palace 1 in Beijing, China, with looking at these closed habitation systems, looking at how people could survive in them and how they interact with the ecological portion of it as well as the psychological portion of it. And then I've been doing this research since the winter of 2014 now, so it's been a few years. And something that's really important that I wanted to bring up in this talk is we talk about closed ecological systems, and it seems like it could be a relatively easy thing. You just build a engineered habitation system around an ecosystem and it should work, right? Well, there's actually problems that we're seeing with closing off ecosystems, and a great example that is commonly used is the problems that Biosphere 2 experience, which is currently owned by the University of Arizona. It's where they decided to recreate these large biomes within this structure. You have the tropical rainforest biome, the ocean biome, the savanna biome, etc. To see if you could really close off ecological systems and have people survive and live in that. And they started to see some issues with this. They saw that there were some atmospheric fluctuations and introduction of volatile organic compounds in that combined space. There was uncontrolled biological activity of microbial populations due to the influx of a lot of organic matter and the decomposition that was happening. And these atmospherics and what the microbes were doing was also interacting with the materials that they used to engineer the system. The cement that they had used was also affecting the atmospherics and interacting with the system, with the ecological system. And so you can see that when you try to close off an ecological system, there's biogeochemical cycles as well as other conditions that will be affected by this. So we have to understand that closing off ecological systems is not as simple as it may seem, and that there's a lot that we need to consider, from the microbial scale to the atmospherics and how everything interacts together. We need to understand these things to understand how we can mitigate and prevent these problems from cropping up when we have a closed ecological system on the moon or Mars. And so agricultural systems, no matter if they're on Earth or in space, you need to have some considerations. You need a system that is stable, resilient, sustainable, adaptable to any unknowns that come in, like an unknown biovector that comes in, changes in climate, a disease that comes in and wipes out all of your plants, things like that. And then you also have to consider, is your system going to be independent, or is it going to rely on a politically and financially expensive supply chain? So there's a lot to consider here. And so a lot of my research that I've been doing for a couple years now has been looking at how can we redesign closed ecological systems. And I actually have a patent now for something that I retermed a quasi-closed agroecological system. And to tell the difference between what a closed ecological system is and a quasi-closed agroecological system, here's the two definitions. Pretty much the major difference is that with the system that I've developed for quasi-closed agroecological systems, we're considering ecological theories and principles and processes, and really getting down into how we can use the in situ resources in the area that we have these systems. And so this is just a visual comparison of what a closed ecological system would look like with the Soviet Union's BIOS experiment, and then a quasi-closed agroecological system that my company designed. And so you have to consider agroecology with these systems, which is applying ecological and social concepts and principles to the design and management of agricultural systems. You need to consider food security and the dimensions of food security. Are you going to have availability of food, access to food? How are you going to use the food, and the stability of the entire system? You also have to consider that quasi-closed agroecological systems go beyond just providing food. They provide services, ecological services, to your system itself through regulating soil formation, habitat photosynthesis, regulating services through climate regulation, but also cultural services and the psychological and mental connection that we have with our environment. And a lot of my research focuses on the soil formation side, which really gets into this theory of ecological succession. How do we go from bare rock, like what we would have on Mars, to these beautiful formed ecological systems that can survive disturbances? Like if a fire comes through here on Earth, we see that the forest that was affected by that fire is able to rebuild itself and come back. So how do we go from bare rock to systems that are able to be resilient and sustainable? And so we're starting off with these they look really degraded, and they're pretty much sand and dust, these geological substrates, and we want it to look something like this. And what's interesting is my research actually looked at changing this Mars regolith simulant into this, and that's actually the initial and final product of the research that I did. So you can see that our understanding of soil and how we can revitalize it for sustainable agroecological system use is possible, and there's definitely a lot more research that needs to be done here. And soil is so important because 95% of the food produced around the world here on Earth is directly or indirectly produced using soil. So we already rely on it very much for the majority of our food supply. So we really have to consider it when we're thinking about space agriculture. And some of you may be thinking, well, is this even possible? Well, we can look back at traditional agricultural systems and the history of agriculture and see that there are civilizations out there that have been able to produce very biodiverse, thriving agricultural systems in desert environments, such as in the Negev Desert and hinterlands, which is where the Nabatean civilization was founded, where they were able to support 30,000 people at their peak in that kind of environment, completely food secure. So it's very important that we also look at our history and our agricultural systems and our traditional agricultural systems and see the best practices and the sustainable practices that they've brought forward. And so my current research is being done at Cornell University as a student. A lot of the previous research I talked about was at Duke University, as well as with my company, Deep Space Ecology. And the research that I'm doing focuses a lot on the soil microbiome and looking at microbial adhesion to organic matter and minerals and really understanding what's happening at the adhesive mineral and organic interface. So really getting to the chemistry of how do microorganisms adhere in the soil and how does this affect soil aggregation and soil organic matter decomposition and the persistence of carbon. And then the space side of my research is actually sending soil and microorganisms into space, which I have an international space experiment coming up. And so, yeah, that's also looking at dynamics and looking at how, if gravity is really part of these stabilization mechanisms for microorganisms. And I know that I'm running out of time, so I will just skip that. And here's my social media. Thank you so much for your time.
Thank you, Morgan. So if things are unclear, please leave some questions in the Q&A, and I will try to answer some questions right now. Morgan, what really popped up for me is I personally didn't know that there was a mission to Mars in 2024 that would be human-manned. That's really surprising to me. And secondly, you mentioned that even manufacturing materials can have an effect on closed ecosystems. So let's see if I have questions popping in. If not, I'll ask one myself. I don't currently see questions, so I'll ask a quick question. So if that's the case, so do you imagine like making building materials out of like Mars soil? Is that then also a priority, do you believe? And is maybe some of your research working on that too?
Yeah, so when we're thinking about what we're going to need to use, you definitely have to use materials that will be able to pretty much seal off your atmospherics because you're going to be in this extreme environment. But the reason why I call these systems I've developed quasi-closed is because we have to consider the in situ resources of your area and how you can use those resources in your system and have this exchange happening. Which is something that we do here on Earth as well, using our local resources. So we need to make sure that we understand the resources that we have on the moon, on Mars, that could be used potentially, not necessarily for actually building the most important parts of the habitation system, because we need to make sure it's habitable and safe for people, but with using like 3D printing and using like the Mars regolith, you can design landscapes, you can design different aspects of the interior build, as well as covering your system if you want to have some radiation shielding. That's also something that's being considered as well. So yeah, you have to consider your in situ resources that you're going to be using, but also how your ecological system is interacting with your technology, how the microorganisms interact with the materials you use, how the atmospherics interact with the materials you use, making sure there's no unnecessary oxidation that's happening, things like that. So this is why interdisciplinary teams are so important. So you have these conversations and make sure that you do these risk mitigations and make sure you're using everything in your power to make it as safe for whoever's going to be living in it.
Fantastic. All right, great. Thank you so much, Morgan. So the other questions that came in, I will ask them during the Q&A later on. So let's move on to our next speaker. Our next speaker, let's see here, is Marie. So Dr. Marie Fuseau is a professor at the Department of Food Sciences, ULaval University. and her research is mainly focused on microbial interactions, just like Morgan, but here both bacterial and fungal, and in the context more of food quality. So during her doctorate, for example, she studied maple sap microbial ecology in relation to syrup quality. Such a fascinating research. So Marie, you can go ahead and share your screen.
Hello. Let me get it to the presentation. Here. Is it okay for everyone? Thank you. All right, so thank you for the introduction. So I'm going to talk about something totally different from the previous speaker, more on the side of, well, of food, just a bit of scientific of my scientific background. So I'm a trained microbiologist, and then for my graduate studies, I went to the side of food science. So I wanted to work with the good microbes instead of going with the pathogen side. And I was kind of a black sheep in my lab because where I was, everyone was studying cheese, but I chose to work with maple syrup. And maybe the link between microbes and maple syrup is not that obvious, but I can show you a bit later why it has such a great importance. So some of my friends say that I have the most Canadian PhD in the world because I did it on maple syrup. And after that, well, obviously it's kind of a very niche topic, maple syrup. So I had to go and do something else. So as a postdoc, I went and worked at the IBIS Institute that some of you may know. I worked in evolution and systems biology. So work with fish and also with yeast, and I kind of stuck with those topic a little bit. Now I have my own lab. I'm an adjunct professor at Laval University in the food science department. So I kind of went into a circle. I came back where I was before. And I teach quality in food science. Mostly in my lab, we are interested in how we can manipulate microbiomes, food microbiomes, to, for instance, avoid microbial spoilage. So we are developing methods, for instance, to measure microbial interactions, both between bacteria and yeast mostly. So I can say I am still crazy about maple syrup. It's still one of my favorite topics for research. But we do work also on other types of microorganisms that are, for instance, relevant for the milk or meat industry. So today I wanted to maybe introduce some of you to the world of maple syrup production because it is a very local thing. I mean, here in Quebec, we grow up with these things. We have field trips in school about maple syrup production, but I understand that in other parts of Canada where maple syrup is not produced, such as in on the West Coast, you may not be familiar with how maple syrup is produced, or maybe not with the modern way of producing maple syrup. So there, it starts obviously with a sugar bush. So there are maple trees that get tapped, and nowadays we don't collect sap with buckets. We actually connect the trees with a complex tubing network that is under vacuum, so that the sap can flow faster. And well, one of the things that happens there in this tubing network is that it gets colonized by microorganisms. They actually form kind of biofilms inside of these systems. These biofilms have been characterized before, and we know that they are composed of both bacteria and yeast, but they are very variable from one site to another. And then the next step in this process maybe is to store the sap somewhere, sometimes outside, depending on the setup of the production site. And after a while, I'm just trying to, okay, all right. I had an interfering window in my screen. So when maple sap is collected, like I said, it can be stored outside for a certain amount of time, and then it's going to be directed to the main processing building, where it's going to be stored in tanks that are not necessarily refrigerated. They are kind of inside at the room temperature, which usually is cold because the period when we collect maple sap is during spring, so it's still very cold here in Quebec. And then there are new technologies that have been used to concentrate sap before we have to boil it to concentrate it in maple syrup. So this kind of allows to an economic gain because it costs less to concentrate maple sap, but it creates a little bit of a problem because then you have maple sap concentrate that contains a higher concentration of nutrients. Instead of being around 2% sugar, it will go up to between 8 and 35 Brix degrees. So this concentrate is also very prone to microbial spoilage if you don't do it fast enough. And then this concentrate is then processed in an evaporator. There are all kind of different technologies there. You can still see some producers that use wood to feed into those traditional evaporators, but you can also find electrical ones or ones that use oil. And then the final product is concentrated enough to be stable. If it's packaged warm enough, then there's usually no problem with microbiology afterwards. It can be kept at room temperature, again, if it's not exposed to air. So this is a portrait of how you produce maple syrup nowadays. And this industry has really grown over the years. It was something of an artisanal production up until, I would say, the 1990s. But really in the beginning of 2000, every producer started using these modern system, and then the production really went up, and so producers got organized. They had a federation, so they started to make bulk production of maple syrup. But there are a lot of factors that will get involved in this production process. First, well, quality is usually defined as the color range, so the color intensity, and also in terms of flavor that I will talk about a bit later. So there is obviously a human component involved in making maple syrup because there is technology involved, there is management. For example, you have to keep your network of tubes very clean. So the human factor is important, but there are also other factors such as the chemical composition of maple sap that is very important because you have to have a good material to make a good product. And maple sap is, maple syrup is really a concentrate of maple sap. It's concentrated 35 to 40 times. So even if you have a small contamination of something that tastes bad, if it's not something degraded during the heating process or evaporated, then it will just get more and more concentrated in your syrup, and it's gonna remain a problem. And of course, the tree metabolism is really involved in shaping the composition of maple sap, and it will evolve during the spring period when maple sap is collected. And it also varies because of the age of the tree, because of its health and genetics. During spring, there is this dormancy released process that happens. So the tree is dormant during the winter, but as the temperature gets warmer and warmer and the days get longer, then the tree is starting to wake up, and it will release nutrients at some point in its sap. And then the chemical composition of maple sap is changing during the time period. And this is obviously influenced by the climate and also by the geography. So from year to year and from production site to production site, it's not the same process. So there's a lot of biological variability that is owed to this process. But there's also the famous microbes that will come and complicate even more all of these factors that influence maple syrup quality, because microorganisms that mostly come from the maple syrup microbiome, from the maple tree microbiome, but also maybe from the human interference, So a lot of microbe actually get into the sap, and they can grow because it's full of nutrients. And so depending on the microbial load and on the microbial species, you will see different type of effects that they can have on maple syrup. And that was one of the main thing that I studied during my graduate studies, and that is still a research topic going on in our lab. So maple syrup has a diversity of flavors that can be quite subtle, and probably most of you I've never been exposed to this variation, because most of the products that we can find in stores are actually a composite maple syrup that has been standardized to even out the bad of flavors that can be found. The classification system is based on two aspects. First, the intensity of a flavor. If it's standard, it's okay, but it can have off flavors that can be light, moderate, or strong, and depending on those, it's going to get classified differently. As you can see here, the yearly percentages of problems in terms of flavor can be quite high. The light of flavor problems are usually easy to mask if you just combine different batch of maple syrup to make larger batches and standardize it. But there are a lot of other that can have moderate or strong flavor defects, and those usually get used for food transformation. For instance, if there is more cooking, then some bad taste can actually disappear. So these on top of the intensity of each defect, they can also be categorized somewhat, but although it's not a perfect system, there are type of defects in taste that are said to be natural. You can have a vegetal taste, a woody taste. There are defects that are from a microbial origin, especially if they were not packaged right. They can be fermentation, post-production of the product, or even the presence of mold. So it can taste moldy, but also if it was not clean enough when the maple sap was collected, there can be residual taste of molds if it were present in the tubing system. Chemical aftertaste can also occur if there are cleaning products that were not rinsed enough, or if the anti-foaming agent was used excessively. There's also a category that is for like free-for-all, because it's not identified defects for kind of everything, or even if it's multiple defects. This is actually really hard to work with in science because it's really not a well-defined category. There is the body syrup defect that is a type of off flavor that we see appearing at the end of the season that comes from an interaction between the tree metabolism and probably microorganism. This is an ongoing research question in my lab to characterize how these microorganisms interfere and help create this off flavor that is really hard to get rid of. And then there's the ropey syrup that is actually a texture defect. And this ropey texture is because some microbial growth in the sap coming from different type of microorganisms, but one of them that has been identified is Leuconostoc mesenteroides, and these produce polysaccharide, exopolysaccharides. And you don't see it when you have the sap, but if you concentrate it, then the concentration of these polysaccharides becomes so high that in the syrup, when you pour it, it actually makes strings. So it's also called stringy syrup. And usually these types of defects, these type of batches are destroyed. So this is kind of a loss for the maple syrup industry. So this is kind of the thing we would like to address. So what I've shown so far to you is that microorganisms can be involved at different steps in maple syrup production. And so far, we know that yeast and bacteria that are found in a maple sap microbiome, they may help, some of them may help actually improve flavor, like Pseudomonas fluorescens has been associated with nice flavors, but it can also cause defects as, for instance, the ropey syrup. So my main questions I have left over here and that I would like to discuss with you is, how can we exploit microorganisms to improve maple syrup quality, or how can we use microorganisms to increase the value of those rejected syrups, I think, which is the project of the iGEM team at ULaval. Thank you. So if you have any questions, I would like to answer that. Thanks.
Thank you so much, Marie. That was fascinating. I really didn't know much about this topic. So let's see if we have some questions coming in. We're also running a bit behind time, so I'm encouraging all of the speakers to help me answer questions by clicking on the Q&A and answering a couple of questions. So let's see. Maybe I'll ask a quick question. So you see this in a couple of other commercial products like coffee, for example, where it becomes more popular to have naturally fermented coffee and naturally fermented products. So I'm wondering if we really work on maple syrup, is there, as you say, a possibility of like fermenting it afterwards, or using specific microorganisms to change the taste and flavor of it? And do you think that is something that would become more popular?
Well, there are some local projects that are also already produced from maple syrup. There are some alcohols that are actually that include maple syrup or some fermentation of maple syrup, and I think it's a great avenue. There could also be other types of projects that could be based obviously on maple syrup. It has a lot of flavor precursors that could actually be exploited. I think it's really an avenue that would be interesting for the industry, but I don't think that there's been very much research done on this topic so far.
Okay, great. I hope we can talk about that during the Q&A. So now let's move on to our final speaker, and then we'll head to a Q&A session. So our last speaker is Professor Karen McDonald. She's a professor of chemical engineering at the University of California, Davis. She is the institutional chair of the NASA-affiliated CUBES, which is the Center for Biological Engineering in Space at UC Davis, and also division leader for the Food and Pharmaceutical Synthesis Division. So Dr. McDonald and her collaborators apply synthetic biology tools, mostly in plants, and try to use them as production systems. So this using the whole plant, harvested tissue, or plant cells grown in bioreactors. And as a biochemical engineer, she is very interested in translational research and strives to develop novel biomanufacturing processes that are scalable, cost-effective, and meet a variety of design constraints. So I'm excited to hear about that engineering aspect in your talk.
Thank you very much. First of all, let me apologize for my eye patch here. I had an accident over the weekend, but I really want to participate in this, so so sorry about that. So yeah, I'm a professor in chemical engineering at University of California, Davis, and also the division lead for the Food and Pharmaceutical Synthesis Division of CUBES, which is the Center for the Utilization of Biological Engineering in Space. It's a NASA Space Technology Research Institute, one of 4 across the country. Trying to figure out how to okay, there we go. So just a little bit about the Food and Pharmaceutical Synthesis Division. We're one of 4 divisions within CUBES, and CUBES is a multi-university center. It involves UC Davis, UC Berkeley, Stanford, Utah State, and University of Florida. And it's really aimed at synthetic biology, developing synthetic biology platform technologies for a Mars mission. And the Food and Pharmaceutical Synthesis Division is really aimed at trying to improve food production technologies, as well as using plants and photosynthetic organisms for pharmaceutical production for astronauts, and as well as recycling of plant waste. So these are the 4 divisions of CUBES. The Microbial Media and Feedstocks Division is one that is going to It's basically focused on in situ resource utilization, carbon dioxide, nitrogen, water, regolith, and other nutrients, as well as light. And then fixing carbon, fixing nitrogen, as well as other nutrients. And then those fixed materials would be used in the biofuels and biomaterial manufacturing division. So that group is looking primarily at ways to produce polymers that could be used in 3D printing processes. We'll also use, in the food and pharmaceutical synthesis division, we'll use nitrogen, fixed nitrogen for growing plants, as well as obviously carbon dioxide and light. So our goal is really food production, as well as production of pharmaceuticals and recovery of plant waste to recycle those materials. So all of these divisions are really integrated, and the systems design and integration division is one that tries to make sure that requirements are being met for each of the other divisions in terms of material requirements. It's obvious that we'll still need to obviously send things to Mars as well, and we're just trying to minimize the amount of material that would need to be sent, and to use synthetic biology to fulfill the needs for food, biopolymers, and pharmaceuticals. So in our division, we focus on using photons for food and pharmaceuticals. Bruce Bugbee from Utah State works on lighting systems and lighting recipes, that is the wavelength distribution to provide to plants to maximize plant growth. Devin Derman-Cole at UC Berkeley works on the plant microbiome engineering, and also engineering plants for improved photosynthesis and improved what we call harvest index. That's the amount of the mass of the plant that's edible versus the non-edible amount. And we work on molecular farming using plants to make biopharmaceuticals. Adam Arkin at UC Berkeley is also working on engineering spirulina for both food and small molecule drugs, vitamin B12 and acetaminophen. So I'll focus a bit more on using plants to address some of the medical challenges in the journey to Mars. And these challenges are a result primarily of the long distance and the fact that you can't really send up medical supplies, no medical evacuation is possible, and there's going to be a communication delay, coupled with the fact that the environment is quite, extreme conditions. There will be space radiation that could have effects on human health as well as drug stability, and the crew is in isolation in a contained space. And there are certain anticipated health challenges that NASA has already identified. There are about 100 different medical conditions that are anticipated, but there's also unanticipated health challenges, things like increased pathogen virulence in space, changes to the immune system. We don't quite know what the pharmacokinetics and pharmacodynamics of drugs will be in a space-like environment, radiation effects on humans or pathogens. There are going to be behavioral and psychological challenges, and even maybe the need for diagnostic kits in this environment. So why use plants as hosts? Well, first of all, we believe that plants and the infrastructure for growing plants will likely be available on Mars, and that's because plants provide food, carbon dioxide consumption, oxygen and water production, as well as psychological benefits. Plants, because they're higher eukaryotic cells, they're capable of synthesizing very complex small molecules. An example here is Taxol, as well as very complex biologic like monoclonal antibodies. It's a simple system. You're using the plant as the bioreactor essentially, and so the plant is able to utilize in situ resources of light and carbon dioxide. As a biologic system, it's self-replicating. You can bring seeds, grow plants to produce more seeds. There's no need for complicated equipment like bioreactors or sterile growth media or power. And plants don't harbor, propagate mammalian viruses. They're generally regarded as safe. We use techniques both for transient production in plant systems, so that's the ability to make something in a non-transgenic plant. It's just a wild type plant, but you can make it rapidly and it has minimal reagent requirements. And there's also the possibility for oral delivery of both drugs and certainly nutraceuticals. So for those of you who aren't familiar with using plants to make biopharmaceuticals, there are two main methods. One is to produce, to genetically engineer the plants so that you introduce the genetic instructions either into the plant nuclear genome or the chloroplast, and that would be stable transformation. And then you could bring up transgenic seeds with you. Or another method is called transient production, where you're basically using the biosynthetic capabilities of the plant to transcribe and translate the foreign DNA. And this offers the advantage that you could have rapid, just-in-time, or on-demand production of unanticipated, to treat unanticipated medical needs. And here you could engineer or on-site, or either bring libraries. And there's lots of different techniques that you can use that are described here, using recombinant plant viruses, recombinant agrobacterium tumefaciens, biolistics, nanoparticles, and other methods. So one of the things that we're working on, which I'll go over pretty quickly, is trying to make a biotherapeutic for bone regeneration. And it is known that there are lots of effects to the body in space, and some of them are shown here. We focus on trying to figure out a way to treat bone loss, which is often, it's known that it occurs both in the International Space Station, but it's also anticipated that in long duration trips like to Mars, that this could be a significant problem. So one of the biologics that we're working on is parathyroid hormone. It's a drug that's used here on Earth to treat osteoporosis. And what we've done is engineered it. So the parathyroid hormone, we're taking amino acids 1 to 34. That's the pink PTH shown on the lower right-hand side. We fused it to the Fc region of a immunoglobulin, IgG1. And that's to increase the circulatory half-life, so we don't have to use it as frequently. It also increases the protein stability and actually makes it easier to purify, because there are techniques that are chromatographic techniques based on protein A chromatography that allow a single step purification in case we need to inject it. So we've got, there are basically 3 different approaches for how you might deliver the PTH-Fc to an astronaut. One is to create transgenic plants, and we're focused on lettuce, so making this PTH-Fc in lettuce. And one approach might be to create the transgenic lettuce and then have oral delivery of it, so astronaut or crew member would eat the transgenic lettuce. Other approaches would be to transiently produce it in lettuce. So there may be lettuce that's grown, and you could use those lettuce plants or leaves to transiently produce the PTH-Fc. You could then either purify it if you needed to inject it, and so that could be through traditional purification strategies, or we're also developing these technologies called plant viral immunosorbent nanoparticles to specifically bind to the PTH-Fc. And the idea here would be these VINs are something that could be made on site in plants. So we've constructed binary vectors for the PTH-Fc fusion protein screened production in a number of different lettuce varieties, selected romaine and crisphead as our the lettuce that we plan to use. We've expressed it, purified it, characterized it, and shown that it also binds to the PTH, the human PTH receptor, so it's functional. So we're in the process of creating transgenic lettuce that produces the PTH-Fc fusion. This just shows sort of the workflow, and we're looking to identify the lettuce lines that have the highest production of the PTH-Fc. For the applications, the amount of PTH that is recommended is 20 micrograms per day per crew member. And so we can convert that amount of PTH to the equivalent amount of PTH-Fc. And right now we're at an expression level of about 12 milligrams of PTH-Fc per kilogram fresh weight of lettuce. Crew members would have to eat about 368 grams of lettuce, so which is quite a bit of lettuce. So we're trying to increase the expression levels so we can reduce that amount. So with that, I'd just like to acknowledge NASA for funding our work and the work of the CUBE Center, as well as collaborators, folks in my lab, and also the Translational Research Institute for Space Health, which is also funding some of our work for this just-in-time rapid production of therapeutics. And just want to acknowledge NASA again, and I'd be happy to answer any questions you might have.
Fantastic. Thank you so much, Dr. Karen. That was really impressive. I love how, you know, you actually consider the amount of end product and the amount of lettuce. That is really nice. So I got a question here from Serena Sino. I think that would be nice to answer. It's probably quite hard to, as you already mentioned in your talk, to assess the effects. Well, you mentioned on the human body of radiation, but what about on the salad? And is there ways that you study the effects of radiation on the plants that you try to produce here on Earth, or do you really have to send up samples to space, etc.?
Well, that's a really good question. There are facilities that NASA has to study effects of radiation or simulated radiation. It's not exactly the same as what's anticipated to be seen when you're traveling to Mars. I think that's one of the sort of big unknowns is, you know, what is the effect of space galactic radiation on both the biological resources that we bring, seeds, plants, people, microbes that we're going to be carrying with us. But there are facilities in that NASA has. I think it's in the Brookhaven National Lab area that they can study simulated radiation exposure. So we haven't done any of that through CUBES, but we are very interested in that topic. I think it's a very important one.
Okay, thank you so much, Karen. All right, so now we'll be moving on to a Q&A session. So for this, you can leave questions for any of the speakers or even general questions. And first of all, I want to thank the speakers because most of you have helped me and answered a lot of questions already by typing in answers. So that's great. We still have a lot of questions to go though, and only about 7 minutes left. So we have to pick the important ones now. And one great one I want to address is from Bettina, came in early in this talk. Would it be possible, this is specifically for Morgan, but I would like to extend it to the other speakers as well. Would it be possible to create a proxy of a closed ecology in a terrarium, for example, at home? It seems like Bettina is working on teaching astrobiology and art to teenagers, and it seems like an interesting idea to do. And so maybe I can extend this question to the other speakers, is like, how do you believe you can transfer your type of research to younger students and teach them about the effects of of microbiology on specific products, because that seems something that's often lost and not a lot of people know about this. So Morgan, maybe you can answer this first. Do you think it's possible to create a closed ecology terrarium at home?
Definitely. Actually, like some of the earliest exposures that maybe most of us have been exposed to is getting one of the soda pop bottles, the liter bottles, cutting them in half, filling it with rock, soil, water, put a plant in there, and then seal it back up. And you have your own homemade terrarium. So that's an example of a technique that I've seen a lot of teachers use for what is a closed ecological system. And you start to see the water cycle in action with like the water beating on the bottle and then falling back down into the soil. So that's definitely something that I would encourage teachers to do, is find a closed container like a soda bottle, and think about all of the resources that you need in an ecological system. What kind of soil you're going to need, how are you going to help with water movement in the environment. The kind of plant that you put in there will also have different needs. It's going to have different water needs. It's going to need different nutrients. Maybe it prefers more sunlight or it prefers shade. So where you put your closed system is also a factor in it as well. So yeah, you should definitely experiment with this and encourage your students to experiment with it, because it's relatively accessible to anyone, especially if you can just go outside, dig up some soil, grab a plant, and pop it into a soda bottle.
Yeah, I've never tried it myself. Okay, I see, Karen, that you wanted to answer a question as well from the audience about PTH in a lettuce. You can go ahead. So the question is, can it produce seeds that can continue to have that trait, or must you have ongoing production capabilities? Sorry.
Yeah, great question, Lee. Yeah, once you have a transgenic plant that produces the PTH, its offspring will also produce the PTH. So yeah, it would be a continuing trait for the transgenic line. If it's a transient expression, generally what you would do is you would produce it and then that would be a one-off. But the transgenic lettuce, which is what we're shooting for, would have it in its seed.
Interesting. Okay. So it seems like we would be able to extend the session until 1:30. So if any of the speakers are still available till that time, could you let me know? And if not, that's also okay, and I will just change the questions. Are you all available still for a bit longer? Or
Yep.
Morgan, I see you. Yes, Marie. All right, that's fantastic. Okay, so the plan was originally that we do a networking session, but I think we can just extend this session here and continue the Q&A because some really, really great questions have been coming in. So that's great. All right, so another question here from Iswajdana. Sorry if I pronounced your name wrong. So what is the probability of microbes or the organisms taken to different planets will have mutations? And I think this comes back again to the radiation question, and maybe also like if again to the previous question, like what if your population or your seeds or whatever, the next generation of that, will that actually be genetically the same, or is it possible that this can mutate due to radiation or effects of space? This can be a question for Karen or Morgan or even Marie, if you've thought about this.
Yeah, I think there are definitely potentials for mutations of microbes. You know, part of it will be how well things are shielded. I think from the radiation, that will be one aspect. But there is the potential, I believe, for that to happen.
All right.
Yeah, and I think it's also important that to see that gravity could potentially also lead to mutations. That's some of the work that I'm going to be doing on the International Space Station with my experiments, looking at how varying gravity could also affect how microorganisms adhere to soil particles and organic matter, and how that affects the soil structure itself, which of course has implications for here on Earth, but also understanding how microorganisms may be evolving on different planets that have a different gravity. So that's also a mutation that we could see as well, about how they adhere to surfaces as well.
I see. And sorry, what is the experiment called again that you will be performing on the ISS? People have been asking about that.
Yeah, so we may have changed the title recently for the experiment, but the title that I remember us having, it's whatever NASA wants us to call it. So the title that I gave it was Soil Microbial Population Dynamics in Soil Aggregation and Microgravity, Implications for Food Security and Agroecosystem Soil Health on Earth. So yeah, it's important to remember that with the International Space Station, even though it's up in space and it's performing space research, a lot of the experiments that are happening up there have to have Earth applications. There has to be research, business applications that go back to how we can improve things here on Earth. So that's something also important to consider.
I see. Oh, there was a nice question I actually wanted to ask all of you, but I think someone already answered it. It was about the lettuce, Dr. Karen. Why was lettuce chosen? Maybe you already answered this, but why not a more nutrient dense, like a potato, for example?
Yeah, so we chose lettuce because NASA and others have a lot of experience with growing lettuce for space applications. In fact, on the International Space Station, it's actually been used in the past to produce other biopharmaceuticals. So it's sort of not the most well-studied, but a fairly well-studied host for therapeutics production. But the techniques that we're using, as I mentioned, it could be applied to different plants as well. We focus a lot on green leafy plants, but it could be spinach. So the techniques are generally applicable, but we wanted to pick a plant that there's a lot of experience already growing in space-like applications. So.
Okay, yeah, that makes sense. And maybe I want to give the word back to Marie. We haven't heard from you that much since it's mostly about space. And I mean, it would be fantastic to have maple syrup in space, but growing trees might be harder in space, I assume. And maybe Morgan and Dr. Karen can comment on that. I'm just imagining that, well, you have deeper roots, a tree grows tall, and just thinking about gravity, that might just be a whole other problem that I don't know much about. So there's a question here about, again, the effect of microbes on maple syrup taste. I think you mostly addressed like the negative effects of certain microbes on the taste of maple syrup, but maybe you can again maybe talk about some of the positive effects.
Yes. Well, actually, maple sap is just like a culture media for microorganisms. I mean, if you wanted to have maple syrup in space, even then, I don't think that you would necessarily need the tree. I mean, at some point there may be a way to make synthetic maple syrup from something else, as we are discovering what happens. One of the things is that we still don't know exactly what is the maple syrup taste? What is the molecule behind it? We have an idea because it's kind of complex. It looks like it comes from many molecules. So there are on the market some synthetic flavors of maple syrup, but I don't know if you guys have tried them out. It's not like the real deal, but I think as we figure out how microorganisms are involved in developing the maple syrup taste, because I can say that making maple syrup from sterile maple sap, it doesn't taste good at all. So it kind of needs to have microorganisms there. But you know, their biomass, when you heat up maple sap, it's also included in the maple syrup. So it's like their own component may contribute to maple syrup flavor. So there's a lot going on that still has to be uncovered. But I think it's one of the things that will get studied in the future about how we can really put our finger on how to make this taste, and maybe eventually it's something that could be produced for space application if you want to have something really tasteful. May I direct one question to the other panelists, because I can't do that in the Q&A. Yeah. So well, I wanted to know if you've thought about making food from yeast, actually, because like nutritional yeast, maybe instead of using plants, or why not use microbes? And the other part of that question would be, well, we've talked about mutation and the evolution that can occur in space. I know that on the time frame, maybe of those missions, it's like obvious that there is going to be some kind of evolution, but from the on the microbial side, maybe I'm not on the lettuce side, but how do you expect to control that?
I guess I could answer about the yeast. So parts of, there are teams in Cubes that are working on spirulina as a potential food source. And I think our focus is trying to use photoautotrophic organisms that can multiply using light and carbon dioxide, so that we don't have to, you know, bring up you know, traditional bioreactors and have nutrient sources to grow organisms. So that's been our focus, but I can see that yeast, you know, could be a valuable food source as well. And the other question was, what was your other question, Marie?
My other question was about the evolution of microorganisms in there. How do you factor that in your, for example, in your closed system?
Yeah, that's a really good question. Yeah, what we're trying to do, or some of the groups in Cubes are trying to sort of evolve a plant microbiome that will be sort of supportive for growth of plants in a Mars-like environment. And so that means, you know, high CO2, and under the lighting scenarios that we anticipate with photovoltaics, we anticipate that the plants will probably be grown hydroponically with LEDs driven by photovoltaic sources. So, but whether they evolve further once they're implemented, that's a very good question. You know, we're trying to design these communities, microbial communities, based on experiments here on Earth, and yeah, they could evolve and change. So that's a really good question.
I guess making them pre-adapted before sending them out is a good strategy.
Yeah, and trying to see how stable they could be, at least in simulated Mars-like environment here on Earth.
Thank you.
Exactly, or you could see maybe directed evolution approaches, as often done in synthetic biology, exposing them to radiation here on Earth and things like that. Yeah, interesting, fantastic. Maybe we can end on an ethical question, since we're talking about inoculating Mars, I guess. What are your opinions on this? Should we, I mean, as a human race, I guess, if we're going towards this question, can we, maybe at some point, this will be something that's really necessary for us to survive or to thrive on a different planet. But when do you think we will have to make this step of looking at, you know, growing microbes on different planets? And when do you think we will consider this ethical, or maybe we already do?
I guess I can go first. So there are currently planetary protection laws in place that were put in place by the United Nations. And pretty much these are to prevent the contamination of other planetary bodies. And this can be very important, especially if we end up finding microorganisms on Mars. We want to understand that these microorganisms are from Mars and not just microorganisms we accidentally brought with us and didn't realize. And it's also a consideration of making sure we don't repeat some of the same mistakes that we've made here on Earth with the environmental degradation, the extinction of different species. Like we want to make sure that we're approaching having boots on Mars, having a potential habitation system, doing research on Mars. We need to approach it in a very ethical, sustainable way to make sure that if there are residents already there, that we're not imposing on them under the current laws that we have in place.
Mm-hmm. So you said those laws are also work in the opposite direction, that if we take material from Mars and bringing it back here, we might possibly contaminate Earth with microorganisms from Mars. That's an important point.
Yeah, it kind of gets murky with that because we have like we have materials from space that fall to Earth, and so we've brought back like the regolith from the moon. The astronauts had it all over their spacesuits, and so we've definitely brought things back, and things have naturally fallen to Earth that have been exposed to space. So yeah, it needs to be talked about more. I think there's a lot of assumptions that we all make, as well as like hoping that we are able to do what we want moving forward. But I think it's also very important, especially ultimately being an environmental scientist with my background, making sure that we approach this very carefully and have these conversations and just make sure that, like I said, we don't repeat the same mistakes we've had with our environments here on Earth.
Mm-hmm. Absolutely. Are there any further comments on this? Otherwise, we can close off on these beautiful thoughts. I definitely agree. It should be a conversation more often. Yeah, absolutely. All right. So I think that's it. Thank you so much for staying on for a bit longer because we had so many questions in the chat. So I think I have to first thank SAF, which is the Sustainability Action Fund. They have sponsored this event, and iGEM Concordia for running this event. So thank you so much for this. And this concludes the webinar. So yeah, thank you so much. There's still one last poll, I believe, a fun poll for the audience. Which of the following foods is a must-have to eat in space? And we'll post these results on our social media. So this is a fun poll. And I would like to thank Fort Space, obviously, for this event too. So I'll give the work back to you, Anna.
Thanks very much, Kenza. Thanks for moderating and doing a great job handling all these exciting questions coming in. It was very well done. Thank you for the special guest speakers who are available with us today and stayed on a little bit later. We really appreciate your time. And of course, to iGEM Concordia and iGEM Université Laval, thank you so much for organizing this and being here with us today. A recording of today's webinar will be available on our website shortly, which is concordia.ca/4, the number 4. Head over to Fourth Space, check us out on social media at CU Fourth Space to see what we're cooking up next. We've always got great activities going on weekly, so please do follow us and check out our archives for past events such as this one that are available there for you to watch. So thanks everybody once again for being here. We hope you have a great networking session afterwards. We appreciate your time and we will see you next time on Virtual Fourth Space. Thanks iGEM. Thanks everyone. Bye-bye.